there. Why is this showing the cloud thing? Maybe it ended up in one of these other folders. Shouldn't have. Oh, there it is, in the wrong folder. Okay. Take it out. Lab test 1. Okay. I don't know how that happened. Okay. Well, there's about half of you here, which is fine. Hopefully the other half will come to the later lab session, because we are going to start right in on the urinary tract. So remember, on Tuesday is the lab exam, and I did go through everybody's posted the histology drawings that you made, and generally, just as a rubric, I used, if something was not drawn, like one of the slides wasn't drawn for a structure that you needed, then you lost a point. But if I made comments like, this was done mislabeled, it wasn't identified properly, then there wasn't any points taken off. The other thing that would lose points is if there were many things that were unlabeled. So I realized that in the study guide, it doesn't actually say, which is kind of my fault, when we went through all the layers that you were supposed to label along say in the bronchi, all the mucosal layers. And you need to know their tissue subtypes. It's in a slide saying this is what they are, but I did say it multiple times during the lab verbally. I said you should be labeling these. So if it was blatantly things were unlabeled, I took a point off. If there was just occasional ones, some slides had them, some slides didn't, then I didn't take anything off. So we're not talking about lots of points here, but I do want you to realize before you go into the test that if it's visible, you can see it, like the cartilage in the bronchiole. You should know that is in the muscularis externae layer. And I know muscle's difficult to see there, but to realize if you're seeing patches of cartilage, that therefore muscle is in between those patches. And I know we struggle with this with the photos of slides rather than being able to focus up and down, change the contrast that you'd have with a real microscope so you could see these things better. But as long as you know who are the tissue subtypes and you know which layer things are in, I think you'll be okay. So if you're seeing tracheal glands, and I point at those, and I didn't take points off, by the way, if you didn't draw that slide, since the only version of it was in the study guide. It wasn't in the histology photos. But I did note that you didn't know it, so that you make sure that you go into those histology, the pictures that are embedded, the histology pictures embedded in the study guide too. So if I point at those, you should know that's the submucosal layer, and that's typically fibrous connective tissue, and it might contain glands or lymphatic nodules. If I point at the bronchiole and I say, what's this tissue subtype, and I'm pointing at the surface epithelia, then you should know it's pseudo-stratified. You should be able to tell me what zone you're in. If you're in the bronchioli, it's pseudostratified. Then it is, of course, the conducting zone. If I pointed an alveoli, which most of you labeled just fine, but you should know there are type 1 and type 2 cells, so that you're applying what you know about the respiratory system's histology and functions. What do these guys secrete? That kind of thing. Let's see if there was anything else that jumped up. The larynx many times had mislabelings. Again, I didn't take points off for that, but I made in many of your comments for the assignment that you don't see an epiglottis in both the posterior and anterior versions of that photograph. So again, in the study guide, I posted a composite picture where things are labeled so that you can see where the glottis is. And I want to make sure most of you did get this, but some of you didn't, that the glottis is not a structure. The glottis is a space. So you can't point at anything except blank space to label the glottis. And the lining of the entire photos for that part of the larynx is all stratified squamous epithelia. You'd have to be seeing much deeper down in the larynx to see the pseudostratified ciliated epithelia along the glottis. So one other tool in your studying for the lab practical lab test next week is that I did put in the De Fiore's histology book after people were asking, and perhaps that will help you. Remember, a lot of those kind of drawings, the larynx, it will not help you. It's not even a very good picture of the larynx. I think the only tool you'll have for that is what I put in the study guide. So if that helps you, you're welcome to it. I will tell you it's a huge file. So you may wish to either download it to access, at least for me, it can go really slowly. It does have a search function, so you can, look for that one, but that's where I said maybe kind of slow as a use for a reference, because it took me quite a long time to upload it into Canvas. So see how it works for you. Again, this may be useful with the urinary tract, but I do think they're often drawings, but still they can be of use. So hopefully that will help you as you work on the histology. So today we're not going to do histology yet. We're going to be looking at the models for the urinary tract. And again, there are models for the respiratory on the study guide. So make sure you're reading the study guide carefully. Before I leave and go into the urinary system, are there any questions about the lab test or how you want to put those into chat, or you can just speak up? It's either way next week. Okay, it looks like everyone feels like they don't have questions. If you do between now and next Tuesday at 9:20, then you can either ask me in an office hour that Tuesday, or you can email me anytime between now and then. The way the test will work will be the same as last time. This time I will post the answer sheet. I'm still working on it, but I'll post it probably on the weekend. So it won't be as before. There were some questions that were obviously built into the way the answer sheet was worded. It won't be that way this time, so I can post it early. And then if you can, please print it out. If you have no printer, you can type it on an online version of it, a screen version of it. But I would prefer that. Some of you were using pieces of notebook paper, and that's I'll accept it, but it just makes it a lot harder for me to grade it. So you should have it available early enough that hopefully you'll be able to download it or print it. And then again, you'll send me a picture. The test will take a little more than an hour. So we will meet at 9:20, again, during the lecture time this time, and do the test during the lecture time. So we'll meet at 9:20, and I will be doing it by Confer Zoom, sharing my screen with Confer Zoom. And like before, the time you have to answer the question will be at the top of each PowerPoint slide. And then the questions will be on the PowerPoint slide. Again, if you have questions about the test as we're going along, ideally use chat, just not to disturb others. But since everyone's synchronized, in this case, if you want to turn on your microphone and ask a question, you can do that. Yeah, so if you don't have a printer, then I understand. You can even download it as a document and type on it. It'll be a Word document, and then send it to me by Canvas, submitting it through Canvas. Okay, all right, so I'm gonna share the screen. So for a few of you just arrived, while you guys are working on the test, I did upload this. I was slow about doing it only because I haven't really quite finished the rest of the semester. All of the urinary and reproductive's in there, even the beginning, and the acid base, the beginning of the, digestive. But I haven't put all the digestive parts into it. So there'll be, later we'll have more things added to this study guide. So let me share that screen with you. And what my ambition is to do, well, we should be able to do, is also answer the first of the two lecture questions, which are really general questions about the urinary system. So just two of them. So if you want to work on that as well, then you might want to have that out in front of you. Let me get that out in front of me too. When I'm done, you can go ahead and work on your study guide 3, or if you have questions about what you're studying for the lab practical, I'm happy to answer those as well. So this is the cover page of study guide 3. Hopefully you're seeing that. And what I did is I extracted some pictures from the lab manual. Are you seeing these pictures? Just give me a thumbs up, anybody. Okay, great. Sometimes when I switch slides, the stop share doesn't keep sharing, and I think it's particularly, well, it may not be particularly true if I do it as a, let me see. I want to do a slideshow so things will be a little bit bigger. All right, so you have these pictures. I hope you're still seeing them. Anyone, again, if you're not, just give me a thumbs down or something. I can't see all of you. So, all right, great. So it tells you to study these figures and relate them to the models. Some of the photos of the models I have embedded in the study guide, some are not. And so we'll go back and forth. We'll look at the models as well. So it's showing you the entire urinary tract in a female, although they don't have the female structures in here in this particular diagram, so that because they would obscure some of the ureter probably here. So you have 2 kidneys, hopefully you know that, but you are not entirely bilaterally symmetrical with the position of the kidneys in the body. First of all, the kidneys are behind the peritoneal membrane. So if you watch the videos that I post, you'll hear this physician telling which I won't test you on, this particular statement telling you that all the organs of the abdomen start within the peritoneum, but some of them migrate behind the parietal serosa of the peritoneal serosal membranes. And the kidneys are one of them. They become embedded with muscle behind them, but there's very little skeletal structures to actually protect your kidneys. So you may even know in some football games and boxing and things that people sometimes have their kidneys damaged by impact. So to help protect them, and not shown in this picture, but shown down here in the lower picture, you have several layers of protective connective tissues that surround the kidneys. So you only have muscle and, a little bit of bone with your vertebrae to help protect your kidneys. I'm going to move you guys so I can see this whole picture. And those are embedded in capsules. So here it talks about several layers of capsulization around the kidneys that help protect them against impact. And probably the main thing that's helping protect against impact is a lot of fat. So there is a fatty capsule and a fibrous capsule around the outside. And then there's something called fascia, which is again connective tissue, a fibrous connective tissue that binds the kidneys to the surrounding muscle layers that are around the back. So this is the person's back over. Whoops, where'd my pointer go? Over here. This is their back. And here's the peritoneal cavity with the white meaning to be that parietal layer of the peritoneum that lines the walls of the cavity. So you can see the kidneys on each side here. And up here, you can see that the one that is on the left side is just slightly higher than the one that's on the right side. And that's because of the position of the liver taking up so much space, more space on the right side than on the left side. The other thing that is obvious in this picture is that it has the large renal arteries and veins entering in the kidney. We'll look at this a little higher, little bigger picture in a minute. And those actually, this vascularization is separate from the vascularization of the adrenal glands. These adrenal glands sit within that same renal fascia. So they're sort of packaged together and held together, but vascularization is separate. And so it's almost kind of coincidental that they're sitting together within the same packaging. But as we'll see, the vascularization of the kidney, we're going to study that, and just know it's separate from the adrenal glands. Okay, the ureter exits the kidney in the same region that the blood vessels enter. And so this indentation gets reinforced with some denser connective tissue, and that's called the hilum again. So you have a hilum for the kidneys, like you had a hilum for the lung, where the bronchi and the large vessels enter the lung. It's kind of reinforced here, where the ureter and the why do I keep losing my pointer? The ureter and the vessels entering go into the kidney. So we'll look at that a little bigger picture in a minute. So here it's saying study the figures and relate them to the models. We'll take a look at the models and understand what it means to be retroperitoneal. The ureter is also retroperitoneal, and it is a mucosal membrane. So you're going to be drawing pictures after the lab test, so next Thursday, of cross sections through the, once again, I lost my pointer, through the ureter. And you'll see that fat that helps protect the kidney is also around the outside of the ureter. And then that's considered part of the adventitia in this case, that is that outermost layer. I don't know why the pointer keeps disappearing, but it does. Anyway, and then you're going to have epithelial layer, a submucosa, and there is 2 layers of smooth muscle around the ureter. So we'll talk more about that when we look at the slides in a week. Eventually, the urine is moved by peristaltic movements plus gravity down to the bladder here. You're going to draw pictures of cross sections through the bladder as well. And it's a mucosal membrane. So we'll go through its layers. And then when you go to urinate, which is also called micturate, you use a mucosal membrane called the urethra. And in the man and the woman, the urethra is quite different, although their mucosal layers are the same. So once again, my pointer disappeared, but here it is. Okay, so we talked about this, understand the reasons for the encapsulation, and the peri means around, the renal fatty capsule, and the fibrous capsule. So I didn't mention the fascia, but you should know at least those 2 capsulizations, and then the position of the adrenal gland and its separate vascularization. Are there any questions? Why don't you just go ahead and speak up, because once I'm showing the slide, it's harder to have a chat here too, if you have any questions about this. All right, so let me look at the next slide for, oh, it's not going to let me do that, huh? All right, so here is a photograph of one of the models that you're going to be studying, and it's one of the ones that shows, it's as equivalent as I can get to the top picture, although it is a man, not a woman, and I'll show you how you know that. And here's a list of things you're going to be looking for. Since this is a man, the way we know that this is a man is it has a prostate here. That's one of the signs. You can see a little bit of the seminal vesicles, but I won't ask them in this view because you don't see it so very well. So here is the bladder, kind of with a reflection on it. Here are the urethras going up. In this case, this kidney has been bisected in a frontal section, but we have other models showing that. And once again, the pointer disappeared. The renal vessels going into this hilum here. In this case, they didn't bisect the kidney. And then this white, brownish, whiter matter up here, those are the adrenal glands kind of sitting on the top. So it's really difficult to see the encapsulation here. So just know there's a fatty capsule. I don't know whether the model, they didn't show it very much. So I didn't put that on that list for this model. All right, we okay with that? Let's see. All right, I want to spend a couple minutes because in 103A you studied the testes and the ovaries and oogenesis and spermatogenesis and looked at slides. We're not going to go back and look at that, but especially for the man, but also for the woman, there are some accessory structures so close to the urinary in anatomy that in the gross anatomy that this is where we're going to have a chance to look at some of the accessory structures that are nearby the bladder and the urethra and how they differ in men and women. So this is actually jumping into the reproductive gross anatomy lab exercise. You're going to be going back and forth. So here in the left side picture, the ureters are coming down into the bladder of a man, and we'll talk about some of the internal structures of the bladder briefly in a minute because they're going to be common for the man and the woman. But with a man, you have these, they're quite enlarged in this diagram. They're really not that big. You have on each side of them seminal vesicles, and they contribute fluid to the ejaculate. Here goes my pointer again. And then you see that when the urine leaves the bladder, it passes through the urethra, which is much, much longer in the man. And along the way, it goes right through the middle of the prostate. So the urethra changes names as it goes. I don't worry too much about the various names of the urethra as long as you recognize, there goes my pointer again, that this is the urethra. So as it passes through the prostate, it's received, urine passes through the prostate and just enters into the penile or spongy urethra and then out of the body when you urinate. Now, when the man is ejaculating, no urine will leave the bladder. We're going to have some sphincters that we'll talk about later, the internal and external sphincters that prevent urine from leaving the bladder when you don't want it to. And but when you are ejaculating, sperm has to pass through the same area, and the sperm, which you can't see in this view, is coming from the vas deferens and stored in the epididymis in the testes. And as it passes through, it has a little bit of testicular fluid that was made in the testes. But most of the volume of the fluid that's going to transport the sperm is coming from the seminal vesicles, and about 30% of it's coming from the prostate. So there are these accessory glands, and we won't be talking about them in lecture, so you need to study this using the study guide and not the lecture. We won't be going through that in lecture pretty much at all, but you can study it in the lab. So there are 3 glands that contribute to what passes through the urethra during ejaculation. The fluid that's transporting the sperm is coming from the seminal vesicles in this prostate. But prior to evacuation, there are 2 little glands embedded in the musculature on each side of the base of the penis that are called the bulbourethral glands. These make what's known as the pre-ejaculate. And so the point of that is to cleanse the long male urethra and adjust pH and make it so that as the ejaculate fluid coming with the sperm in it coming from the other 2 glands passes through the urethra later, that the sperm is not adversely affected by any remnants of urine that might be in the urethra. So on each side of the urethra in the penis, you have erectile tissue. These are labeled here, the corpora spongiosum and the corpora cavernosum. The larger one, which is sort of subdivided by these white bands, is the cavernosum, like little caves. And then on each side, very sort of surrounding it, and we'll see that in another section in a minute, it's sort of like a donut around the urethra itself, is the corpora spongiosum. And the tip of the penis down here, which if I can get my pointer, is all corpora spongiosum. So you can see it kind of continuous along so the head of the penis is all filled with this corpora spongiosum. So what this erectile tissue is a lot of sinus type of capillaries where, or veins and arterioles, rather, not capillaries, where it sort of opens up, and capillaries, I guess, and lots of blood can pool. So why I said arterioles and venules was because you need to vasodilate those in order to cause blood to pool within the capillary beds that make up this vasculature of the spongiosum and the cavernosum. Now, why you need 2 isn't really talked about in the lab manual. Why do you have to have both the spongiosum? And my guess about that is that when the vasodilation occurs, you want this one that's kind of a donut around the urethra to help maintain that the urethra is still open as the rest of the corpora cavernosum swells on each side of it, so they don't swell to the point where they're closing off the opening of the urethra. So for the man, we'll look at this a little bit more in the next few slides, but over here we have the woman. You can see the urethra is much shorter. You don't have any, you don't have eggs or any gametes passing through the same passageway like you have in a man, but you do have an internal and external sphincter, and the urethra is nearby the opening of the female reproductive tract, the vagina. So in other other models and pictures, we'll see those as well. So we'll come back to this, but you see in both the man and the woman, the bladder has a region that's called the trigone. So that's just describing where the two urethra openings are, or ureter openings are, and the single urethral opening. So the tri is because of these three openings. The significance of the trigone is that if a bladder infection begins, it's often this region, sorry, there goes my pointer again, this region that gets most inflamed. This is a mucosal membrane again, and the reason why that is significant is if you have just a bladder infection, which is usually as far as it goes, then the infection is limited to the bladder. But because this area has the openings to the ureter, it is possible, if things don't go your way, that you can have infectious organisms go up the ureters. They would have to be going upstream against the mucus that's coming down, and eventually cause an infection of the kidneys, which would be a more serious infection. So that's where since it's labeled, I'm just giving you the significance of the trigone. The other thing you see within the interior of the bladder, and you'll see this in our models as well, is a structure, you're going to hear this term in the digestive tract as well, called rugae. So rugae means that the surface is not smooth. It has little mountains and valleys. So we won't really be able to see that in our histology sections of the bladder, but understand in the models that that's what you're seeing when you see dark and light areas. That, since you're only seeing photos of the models, you would see in a real model that it's not a smooth surface. So the rugae are there partly to help when you go to empty your bladder, to help efficiently empty the bladder, kind of like the trabeculae were in the heart, to help be more efficient at emptying it. So the next two are mid-sagittal sections through the male and the female, showing both the bladder and the reproductive organs that you need to be able to identify in now mid-sagittal section. And these match really pretty well with the pictures in the lab exercise in your lab manual. So you should be able to identify the list of structures that are asked for in the study guide. So for the male, again, here's the bladder with the rugae. I think you can see this looks like it's not smooth. You actually do see a little of the ureter, but I think I would ask it in the other one since you can see much more of it. And then you can see the urethra passing through the prostate here. Now, this little side channel is where the seminal vesicle supplies its fluid into the testicular fluid. And then the bulbourethral, I don't think is particularly visible here because they are on each side of the mid-sagittal. They're not actually really visible here, but they are visible in that previous picture. So I would know they exist, even though they're not really, as far as I can tell, shown in this. And that would make sense since they're not right on the midline. It wouldn't be cut through. This is actually, my pointer would show up again. This is the pubic symphysis. This is bone here. So remember that symphysis is where your two pelvic bones come together and have that fibrocartilage from 103A. And then here, this with the lines is the corpora cavernosum, and the little ones with, looks more pink, I guess, is the spongiosum on each side of the penile urethra. And then here's the head of the penis with spongiosum in it. The testes are here, and they're not showing you the vas deferens because, again, they would be not mid-sagittal, but they are showing you, I think that's what this is meant to represent, is the epididymis. So the epididymis is where sperm is being made constantly, and it's stored in the epididymis until an ejaculation occurs. The last structures, because we will come back to this later, are the anus, and we'll talk more about its large intestine, the last part of the large intestine, but we'll talk about that in the digestive tract. The main reason I still have it on the list is, just because of the difference in men and women, where the anal opening and the urethral opening in the man are really far apart. So having bacteria from the anus, which don't normally reside along the urethra at all, being able to infect the urethra is pretty difficult in a man, but it's pretty easy to happen in a woman, which is one reason why women tend to have more bladder infections than men do overall. Let me move you guys again. I wonder if I can move you over. Nope, I can't. Okay. Because I can see most of this model, but not all of it. So notice the bladder in the female, and why it's looking a little flattened is because what sits above it is the uterus. So this is, again, mid-sagittal section, and we will talk about the layers of the uterus, but you drew this in 103A. So you have the endometrial layer, the myometrial layer, and this is actually part of the peritoneum, but it's called the epimetrium as well. Here you see the fallopian tube, or the uterine tube, sometimes it's called, and it's little fimbriae at the end. We're going to see better pictures of that, where it's hugging the ovary. So the urethra, much shorter in the female. Here's the pubic symphysis, and the vagina running just behind the urethra here. And we will talk a little bit about the other structures like the cervix of the uterus, which really is only just part of the uterus itself. It's not a separate structure. There goes my pointer again. And here is the rectum and the anus. So you can see it's much closer to the urethra, which is why. Now I've got to move you to see these guys, which I don't know why I'm not able to do. But it's hard for me to, maybe if I put it up here, there we go. You can see the two lips. Labia means lips. So you have a labia minor and a labia major. The labia major out here is normally covered with hair and not shown. No hair on this guy either. But it is covered with hair and normal skin layers. The labia minor, the inner lips, which my pointer disappeared again, there it goes, is not covered with hair and has a much thinner layer of skin over it, but both of these close the openings of the vagina and the urethra in the female. You don't have anything really equivalent to that in the male. So let me go to the next The next slide. So here are the equivalent pictures in the lab book. This one is showing you the bulbourethral gland, even though it is actually not found or seen in a mid-sagittal section. It's a little off from the middle of the body. So it's good reference. And here, this cross-section through the penis is showing you why if you enlarge these two corpora cavernosum, so they started swelling, they would swell downward and potentially close the urethra. These are blood vessels in the middle. We don't have to worry about that. But what's in the middle here is the urethra. So by having this donut of the spongiosum around the outside that's swelling, away from the urethra, you're pushing back. Again, it disappeared. You're pushing back at the swelling of these two cavernosum and keeping the urethra open during an ejaculation. This also shows you better than you could see in our models the vas deferens, ductus deferens, which you don't actually have mid-sagittally, and the seminal vesicles here, which are making the majority of the fluids of the ejaculate. So here are the list for the male that you should know the functions of. I haven't asked about histology really along here. With the urethra we will, but not for the seminal vesicles. I mean, they are glands, but I wouldn't ask their histology because we're not doing any sections through any of that stuff. So study this from the lab manual. And I think here is the female, again, quite similar. So I didn't include the vestibular gland or the clitoris. I don't think the clitoris is on here. Oh, it is. Clitoris is on here, which is the rectile tissue of the female, very similar looking to the corpora cavernosum in the male. So here's your list of what you'll be looking for, the functions of. And only other thing I will say about this probably is that there are a number of ligaments you're going to be learning in the female. So the uterus is being suspended from the peritoneum by quite a few ligaments. You see two of them here. I don't think I have this one on the list, the uteral sacral, but I have the round ligament over here, the broad ligament. So it's almost like the uterus is suspended from cables from the peritoneal body wall. And hopefully that makes sense to you because of pregnancy. Because when you're not pregnant, then the uterus is quite small. It doesn't need a lot of support. But once you become pregnant, you need to support this fairly heavy organ within your body. The other thing that's kind of misleading in this picture and in our models is it doesn't include all the other abdominal structures that are in here, like all your intestines. They say the uterus is about the size of a walnut in a non-pregnant woman. Obviously, it's much larger in a pregnant woman. You can also kind of see why you have to urinate a lot when you're pregnant, because as this expands, it's going to be pushing down on the bladder even more. But these ligaments that you're going to be learning help to suspend it and support it when you're pregnant. So I think I have on here the round ligament. Let me see here are the other ones that you'll learn. This thing is called the broad ligament. So this is actually attached to that peritoneal body wall on each lateral side. It's missing on this one, but it has it. So you can see that not only helps hold the uterus as it grows in place, but it also helps hold the ovary in relationship to the uterine or fallopian tube and the fimbriae of the part of the fallopian tube that kind of hugs the ovary. Now these fimbriae have a ciliated cilia on the outside. And what they're doing is moving the serosal fluid of the peritoneum continuously through the uterine or fallopian tube, and then continuously through the uterus and out the vagina. So when you're pregnant or not pregnant, whichever, you have this flow of fluid that's going constantly through the female reproductive structures. These are all mucosal membranes, and there's mucus being secreted along the surface of the vagina especially, but even along the surface of the uterus to help trap organisms and make an outward flow to flush the reproductive structures of the female in a continuous way and prevent infection. So if you get infection of the uterus, the fallopian tubes, this is a pelvic inflammatory disease is often the category it falls under is pelvic inflammatory disease. And if that infection spreads up into the fallopian tubes, it can lead to sterility in the female because the fallopian tube opening is extremely tiny. I think the book says it's the size of a human hair. So any scar tissue buildup from infection within the fallopian tubes can make it so that the egg can never make it to the uterus. The sperm are a lot smaller, so sometimes sperm can get through and fertilize the egg somewhere else, like earlier in the fallopian tube. The embryo will embed in the wall of the fallopian tube just as easily as in the wall of the uterus. And this can lead to a life-threatening condition called an ectopic pregnancy. So ectopic means in the wrong place. So if that baby or embryo embeds along the fallopian tube, it will grow. And for at least a while, it generally won't make a placenta, but it will grow on its own for a while. And this is a fairly small structure. So as it expands, it could cause rupturing, and a woman can bleed to death from that. So the main reason, you can tell you're pregnant with a pregnancy test that you want to go in early in your pregnancy and have a check by a physician is that they're checking to make sure that pregnancy has actually implanted in the uterus and not in the fallopian tube. It can even implant, for that matter, out here in the body wall. It won't go very far. It won't make a placenta, but it's good to know if that happened. It would probably be reabsorbed eventually. So again, we're not going to do other than recognize the ovary as the ovary, but know these structures and the functions of them. Don't worry about all these subcategories of the uterine tube, just the uterine tube. So once again, there is a list over here of things that you're learning. So besides the ligaments that hold the uterus, you also have a ligament that holds the ovary in place and helps to suspend the ovary. It's embedded in this broad ligament, but it has an ovarian ligament. And those are the three ligaments I have for you to learn in the female. So here is a picture from the cadaver pictures that are on mastering. And I believe you have access to these as well. So I may use one of these pictures. It looks a little bit like a crab or something here, but that's the uterus with its ligaments. And I thought I put this in here in part so that you have an idea of how it sits in the body and its size in the body. So you could see if I can get my pointer to show up again, you can see the bladder down here. And the uterus has been kind of pulled up away from it. Normally it would rest lower, so they could show you the ligaments and the uterus itself. And then the ovary, which people don't really think about, but the ovary is on the posterior side of the uterus. It's sort of hidden behind the uterus, and you can see both ovaries there. This is part of the last part of the colon that's going and diving behind all these structures. So take a look at the cadaver here. All right, so I think we will look at, well, we have two ways of looking at the models and the drawings. This is right out of your lab manual again. This is the kidney bisected. And again, there are lists of things that you need to study and know the functions of, and in some cases, the histology of. So here's that ureter coming out of the hilum, that reinforced indentation where the blood vessels come. The urine collects in these spaces, which are called calyces. I think I spelled that right. The major and minor calyces. So the major ones are the larger, wider openings. It's just like changing a road name. And they lead into what they call the pelvis here, which is where all the major calyces come together. The minor calyces are up here between what are called the columns, and at the base of these areas called the pyramids. So these somewhat triangular areas are called pyramids. So here is a kidney that's been bisected, that's a real kidney. I think it's a sheep kidney in this case, but you can read the book, what do we call the figure legend in the book to see if it's a sheep kidney. Now, I posted a dissection of a human kidney, and it's a video that you should watch where they do bisect it, and he does explain these structures, so you can watch that on your own. Now, the kidney in general is divided into a region called the cortex on the outside, and all of these pyramids and columns are part of, and the calyces are part of the medulla that's underneath. So once again, we're going to have a cortex and a medulla like you have in say, lymph nodes or the thymus. And it's just meaning a sort of outer tissue and inner tissue. So study these structures here, including being able to recognize blood vessels. So I probably wouldn't do it in a picture like this to recognize blood vessels, because you'll see I have models that show the blood vessels pretty well. But at least you have them labeled in your lab manual. Okay, so here's a cadaver. Here's the link to go and watch Dr. Youssef's human kidney dissection. And it looks like some student probably videotaped it, but I thought it was a good sense of what the kidney looks like. But also these, I'm fairly sure are cadaver kidneys, but you don't get the sense of the actual size of the kidney from here. You do in the video. And he does walk through and talk about the various structures. So these, again, I got off of mastering these pictures. So we will, let me see what I planned. Yeah, we will stop here. And I'm going to stop sharing and address the first two lecture questions. And then maybe before we do that, I will stop sharing that and close. Let me escape this. And I'll close this. And I will show you in the urinary models a few. This is all on in your lab test 3. So let me open up. Which one do I want to do? Yeah, not that one. Okay, well, we already looked at that one. I guess we'll look at this one. Let me turn it. Okay, let's share the screen. There's quite a few models of the kidney. Let me pull you guys over so I can share it. And here's one of the larger ones of a mid-sagittal section through the kidney. So once again, you have the ureter here. Hopefully you're seeing this. And that leads into this common road of the pelvis. And then you have these wider ones where two things are leading into that, called the major calyces. The minor calyces are what leads into that. Those are all draining urine, they're receiving urine from these pyramids. So we will go into what's in the pyramids. This model also shows you the blood vessels. So here's the renal artery, the renal vein. Now, the arteries and the veins, like the other arteries and veins that you were studying in the blood vessel, by and large have the same names. There is one exception. As the arteries come in, they divide into what are called segmental arteries, whereas the veins, for once, have fewer veins. There are no equivalent segmental veins. I'm not sure why they don't call these segmental veins, but they don't. So you go right from the renal vein into the interlobular veins. They're in between the lobes here. And same with artery. The artery's interlobular. It's segmental down here. And then it becomes an arcuate artery in a vein. See, it's an arch that then branches off to the cortico. This is the cortex part. Cortico-radiant, they're radiating around the cortex, and eventually brings the blood into the repeating structure of the kidney called the nephron. So we're going to start next time looking at the nephron, and it's when you get to the nephron, you'll be seeing histology pictures of the kidney. So we're going to end up drawing histology pictures in the cortex and in the medulla, and there's going to be several not only several versions, but you're going to have to make several drawings in order to see all the structures. Okay, so that's next Thursday after this lab test is over with. So I'm orienting you. Obviously, we can come back and go through more detail if you have or questions, so that you know where to go in the lab manual and in here to look for things. All right, I'm going to stop sharing this one. And let's just talk about the first two lecture questions. And I'll leave you at that. What time we got? We got time for you guys to work on this when you're done, or to ask me questions from respiratory if you need to. So let me open up. New. So I know you may not all have them out, but if you have your lecture questions available, let me share this. I don't know why it does that. So the first lecture question asks about 6 functions of the kidney in addition to producing urine. So we have one, it produces urine. And I'm not going to try and get you guys to work on all those other functions because they are outlined in the book well. But I just figure we need some broader context, and it also brings back or touches on where the lectures are going. The kidney and the urinary, not so much micturition, that's not very hard, or the urinary, ureter and bladder, but really understanding urine production and all the other functions of the kidney is kind of a challenging part. Some of these things you've touched on before. You kind of know what some of the other things the kidney does, like it releases renin, right? And it does that because it acts as a baroreceptor. to help you maintain as a baroreceptor for blood pressure. So this goes back to our first test. So that's one thing you're already aware that the kidney does. You may also remember it releases EPO. So it acts as both the control center and the receptor for the negative feedback regulation of red blood cell production. So I'll just put RBC regulation via negative feedback. So that's all stuff we've done in the past. What we're going to do after we finish the kidney is we're going to look at water balance, salt balance, and pH balance. And the kidney plays a role in all of those, some of which you already know. So water, blood volume balance, meaning it helps maintain blood volume and water balance by responding to ADH and either concentrating the urine, or if ADH is present, so this would concentrate the urine and increase blood volume. It also can regulate salts. And this is, again, some of it goes back to 103A. Salts are electrolytes, same thing. So, for example, aldosterone, leads to an increase in sodium, but it also decreases potassium because it's actually going to act through the sodium-potassium pump. And again, back from 103A, PTH is a regulator of calcium, and as we'll see, it increases calcium, but it then decreases phosphates, PO4 plus, I believe. Is that right? Phosphates. So salt balance and water balance by the body. The kidney is an effector in this case, and it does pH regulation. So we started alluding to that in the respiratory system. It's going to work alongside the respiratory system, so we won't leave the respiratory system entirely when we go into looking at acid, base, and pH balance. And one of its main roles, or it kind of does this two ways. One, it reabsorbs of bicarbonate, and it can actually make new bicarbonate. It can also increase bicarbonate. And it's the only way that you can actually secrete H+ from your body. So again, this is foreshadowing where we're going before the next lecture test. We're going to talk about the kidney. Basically, almost the whole time, we'll talk about some other organs as well, but it's really going to be all about the kidney. So we have another one, actually, even though I only asked for 6 of them. But this, again, is review from 103A, a million years ago. You might remember it also as maybe as part of the salt balance is how I did it. It takes vitamin D and makes it into calcitriol so that you can absorb more calcium from your small intestine. So lots and lots of roles for the kidney, that some of which we've touched on and some of which we haven't. One other thing that it plays a role in is blood glucose levels. Now, partly because you have to reabsorb the glucose that might otherwise be lost in your urine. And that's the main way you'll see it, but it has some special chemistry that we'll talk about that it does in order to help maintain those blood glucose levels. So we'll come back to that more later. All right, so those are more than 6 roles that the kidney does. And we'll be diving into how, mostly focusing initially on this one, how we're making urine. But as we're making urine, we're going to have to reabsorb salts, water, so that good stuff is maintained, glucose maintained in your body. And we'll talk about that next time. So that's question number 1. Question number 2 is about what we just covered in the lab slides. So it asks you to give a function, hopefully you can do this now, a function for the kidney capsule. So there's really 2 capsules, right? There's the fatty capsule, there's sort of 3 with the fascia, and then there's the fibrous capsules. So this is fibrous connective tissue, this is adipose. The cushioning is kind of a cooperation between the 2, but adipose does a lot more of that. The other on the list, as we go through the lab, I think you'll know more why you have a cortex versus the medulla's pyramids. Different parts of the nephron are located in these different areas, and there's a salt gradient within the pyramid. So we will come back to that more. I already talked about the calyces, the pelvis, where the urine is collected within the kidney itself and sent into the ureter. The ureter then can move that urine to the bladder, both passively through gravity, but also actively by peristaltic movements of its muscularis externae. So I think that finishes up what I wanted. I know it's a lot for a day for an exam, but we kind of fell behind and had to push the exam forward. So that's kind of why everything is being shoved into today. But luckily we do have some labs where you're just working on slides and models, and you can ask questions as you go and look at the models, but do try and use your study guide. If you're finding you're not sure what you have to label or what, please ask during the labs. I'm sitting here and waiting, and we can walk into go in and look at the slides, look at the model photos, and I can answer your questions about that. Okay, so you have about 25 minutes or so left, or 25 minutes. Are there any questions that anyone has? Or do you just need to go and chill for a while? And I will be here. If you would like to come back and ask anything during the next lab section, I'll be here for that too. And some people may show up for that one. And I'll go through the same thing again. Okay, I'm waiting. I'll wait for anyone. You can put a chat question out, or you can just ask me verbally.
I actually have a question about the sport histology for the next exam.
Sure.
Could you walk through the anterior larynx and posterior larynx?
Okay, let me go find those pictures.
Histology slide.
Why don't I'll find those pictures and the study guide so we can go back and forth so you can see. All right, lab assignment 2. Open that. And then I'll come back and open the histology, respiratory. Okay. So let me share this one first. So I don't know if some people just never found this. I did tell you about it, but maybe you never found it. So this is a composite of those two pictures with labels on them, giving you, this is the posterior side, this is the anterior side, and here is the esophagus going along the back, lined with simple stratified squamous epithelia. This structure is where the arytenoid cartilage would be. So it's kind of projecting up. Lots of people labeled this the epiglottis, but it's not. It's the wall between your esophagus and your rest of your larynx. And that vertical piece of cartilage that sticks up, and I wouldn't ask you if that's arytenoid or not, but that's hyaline cartilage right there. So that means that's in that muscularis mucosal layer. This is probably all submucosa that's underneath there, that's in the middle. Then you have this big wide glottis where, as I said, that's a space, that's where air goes. And on the other side is the epiglottis. All of this is still lined with stratified squamous. It's not quite as thick as the stratified squamous of your esophagus, because clearly this is mostly food going through here, whereas this is protected against food that is being directed into there, not being like the food in your esophagus is going to get crushed downward, right, as the esophagus does peristalsis. So you need a lot more layers of stratified squamous, but this is all stratified squamous. This in the middle is the elastic cartilage, and you have submucosa between the two. So this is again in the muscularis mucosa layer. This is all cut edge of the mucosa of your oral cavity. And the epiglottis, like in the models, kind of sticks up. So there's a space behind it. So maybe relate that to the models. So I stop sharing this one, and then let's see the actual individual pictures. They are the same pictures. They're zoomed in a bit more. So here's the epiglottis. You can see the cut edge of the oral cavity. This is all stratified squamous going around the edge. That's where the glottis is. Are you seeing this?
No, we're not able to see.
Oh, what happened? Did I not hit the right? I didn't hit the right picture, sorry. It's the same. Now, hopefully you're seeing it, right?
Yes, thank you.
And are you only seeing the one, or are you seeing, you're probably only seeing the one, yeah. Let me make it bigger then, because it's pretty small. Okay, so here is the glottis with the epiglottis. This is the one that's labeled anterior. This is the cut edge of the mucosa. These are probably maybe even salivary glands under here. Some of it might be fat as well. I had to have to go higher, zoom in to know. And then this is all stratified squamous along the edge, submucosa beneath it. So glottis up here again. Are we good with that?
Yeah, thank you. I think it was just like the orientation of everything.
Yeah, so that's why I put that composite together. So, and if we look at I think more people got the posterior one wrong than the anterior. So let me share that one. Sharing this one. And I know there's just no good sources for this online or anywhere. And the picture they show I don't even know why they show that one in the book. It hardly shows the epiglottis. So once again, this has been cut. So these are cut edges up here. But here is the opening to the esophagus. So as I mentioned, it's almost always pretty collapsed unless you're actually swallowing something. So the two stratified squamous actually almost meet here. If I think I have a picture further down, and it kind of gets wide and you see the gap again. But other than that, this is the esophagus. Again, thicker stratified squamous will be a giveaway. You're looking at the esophagus. So this is the posterior part over here where you have in the middle cartilage right there. And that cartilage right under the pointer is probably the arytenoid cartilage. And this is stratified squamous still with the lining the glottis because you're still up close to the oral cavity. So it's all stratified squamous. Okay, let me stop sharing that. So you have that picture in your study guide that I did add later. It wasn't in the original version of the study guide in case you, and maybe, do I still have the study guide? I want to point out that because so many people made mistakes on that one too. There is a picture in here, it's got labels on it, of the trachea. So I want to point out and make sure that people don't miss that as well. And this is really the only place you see the tracheal glands. So I know they're in the right place, but in the other lower magnification version of the trachea, all you're getting are blood vessels. So there's a blood vessel here, there's actually a blood vessel right here. You can see little red blood cells in it. And then, are you seeing this hopefully?
Yeah, Professor.
Yeah. And the glands are cuboidal cells around the edge. So if I were to use the lower power picture of the trachea that you were drawing, of the cross section, you wouldn't see any tracheal glands. I would just say what is in this submucosal layer, name a structure or something. And these glands secrete mucus, just like the cells along the surface also secrete mucus, so you get lots of mucus. The nice thing about this is you do get a good view of the lamina propria and the pseudostratified ciliated. So if we look at the equivalent picture in your, which one is it? This one? Yeah. So this is the lower power. Stop sharing that. Start sharing this one. Hopefully you're getting, see this one, all these guys, I know this is lower power, we could zoom in a bit. All these guys are blood vessels. You can see blood in them. So you don't have tracheal glands everywhere. Actually, most of the tracheal glands only show up in part of your long trachea where you need more mucus to be released up near the head. If you're down deeper, you don't need as much. There's the pseudostratified. You can really see there's a different layer underneath it that's the lamina propria, like here. It looks redder. And then you're into the submucosa and the hyaline cartilage of the muscularis mucosa. And then this is a bunch of fat or salivary gland, probably fat because I don't see any nuclei in the middle. That's in the adventitia. So lots of people did label the layers on this and only on this. I know I'm talking and talking, but just to point out, some of you were there, some were not in the lab where I think Cal Sang asked me to walk through a few of the other ones. Let me just do one of these bronchioles. Yeah, so this is, let me share this one too, and I'll leave you at that. So you can, as you're doing this over the weekend, hopefully get better at this. Let me make this a bit bigger. So again, cartilage, that's often easy to find, cartilage. Blood vessels are way over here. You're not going to have blood vessels close by like you had in the trachea. Here's more cartilage. If you're seeing cartilage and there are little islands like that, then what's between it that looks kind of patchy over here? That's smooth muscle. So I know at this, if you had the real slide, you probably could actually identify it as smooth muscle. But there is smooth muscle there. No, that's pseudostratified ciliated. That's the lining of the bronchi, the lining of the bronchiole. So pseudostratified ciliated, I would never ask the lamina propria. It's much too low magnification. But if it's right under that, here it's going to be the submucosa. Now, where that line divides from muscle is hard to say, but at least know this is in the muscularis mucosal layer. So this thing is actually a lymphatic nodule, which is quite large here, but I wouldn't ask that one here. And then these are going to be simple squamous. I'd probably use a higher power picture of alveoli if I were to ask that one, though. So that's what some of the labels that we're missing that people didn't have in the, and I realize it's somewhat the way it's worded, it didn't seem like you had to for every time you could, but that is one thing I said verbally to you guys. Okay, are there any other questions about to help you with the test for next week?
Could we go back to that lower magnification of the trachea, the one that you had just?
Okay. All right, let me get rid of this guy and open that one. I think it was tracheal 2. Here we go. And share the screen. And move you guys over so I can share the screen again. Okay, so are you seeing that, Sana?
Yeah, this one. Is the adventitia the layer right directly under the cartilage, like this?
This is actually all cartilage.
Okay.
So cartilage is, this is adventitia here.
Okay.
Cartilage, if you recall back in, especially was obvious in the elastic cartilage that you drew in 103A, it's often got chondrocytes in these lacuna, and then they die and they make these empty cavities. Even if I go up, I don't know if I can go up any more than that. It's kind of out of focus because every time you move a picture, it gets more blurry. So even though it might be in focus as the real photograph, uploading it a couple different times, it just loses. But these guys are where there's the lacuna are really small and they're living chondrocytes, and these are dead chondrocytes in the middle with no cell in the middle of them, or most of them have no nucleus or cell in the middle. There's kind of lacunas that are full of fluid. So this is probably some fibrous connective tissue that, I don't know what layer you'd call it, which is holding the adventitia to the cartilage, but I'd call all of that cartilage. But if I were to ask you, I'd point in the middle here where it's recognizable to you, if that's what you're going after. Okay. Is that good?
Yeah, I think I was just confused as to which layer is the adventitia layer.
Okay. And don't forget the other cartilage picture. Where you can see I saw a lot of people that not a lot, but there were a few people that randomly pointed at something and called it the trachealis muscle. There was more than one picture of the trachea that you needed to have drawn. And I did say that to you when we talked about these slides. So here again, let me make it bigger so here's that hyaline cartilage and the pseudostratified epithelia. And this is in that same layer as the cartilage, but right going across here and attached here is the trachealis muscle. Now, right here not clear to me that's pseudostratified, but I wouldn't ask that because of that. So the trachealis muscle is this area that's kind of a pinker. This is a little oranger that is bridging, and it actually attaches to the cartilage on each side. So remember, that's for causing the airspeed to go up when it contracts, so that when you cough or you sneeze, air will come out of your glottis a lot faster. And it does relax even more than the normal level of tone that might be in it when you swallow reflexively, so that you can move that part towards your esophagus. The esophagus can kind of move into it. I just got distracted by chat. Damn, just can't, like she needs. Oops. You want me to shut up and not talk? Oh, okay. All right. I see it is about something else. Yeah, so anyway, I hadn't been checking chat for questions. If there are other questions, I'm happy to help you with these slides or models. Let me stop sharing, and we're almost out of time for this lab section, but if there are other questions. Okay. All right, you guys, why don't I let you go? Again, I'll be here during the weekend checking my email periodically. Last minute questions, you can get on my office hours at 8 before the lab test, and then the lab test is at 9:20 on Tuesday of next week. So happy studying, and I will start working on the tests and the essays. Don't forget today you're turning in, I have this in announcements, but the essay I emailed you. At first I was going to do it all Ohlone email, but it was very slow going that way. So I did it by going in my regular email. And if it wasn't your Ohlone email that popped up, it was your private, then I sent it there. Many of you just talked to me on email through your private email. So check that one. And if for some reason I'll keep checking up until about 3 today. But if you can't find it, email me soon. I'll check very frequently in my email to make sure you can find your essay, to know which essay you're doing. Okay, I'm going to let you go. And so I will see you next week. And make sure you check announcements. It's got the lists, what you're doing.